Good afternoon, everyone, and welcome to a new EBD seminar. My name is Marijosa Ruiz, and I'm going to be introducing our speaker today. And after her talk, Agus Camacho is going to moderate the questions. A couple of reminders before I introduce our speaker. If you need a certificate of attendance, please write down your name in the chat in YouTube and then send an email to seminars@ebd.csIC.es. Also, if you want to make questions to our speaker today, you can write them down in the chat and then Agus is going to formulate them to the speaker. So our speaker today is Dr. Orly Razgour. She's a senior lecturer at University of Exeter. And those of you who work at the Doñana Biological Station might know her because she collaborates with several researchers here, like Javier Juste. So Orly's research is really, really interesting. We are really happy that she is with us today because she does a very integrative research and she combines ecological, genomic, and geographical information with modeling to understand how biodiversity is affected by climate change and land use change. So without further ado, Orly, you can start whenever you are ready. Thank you so much.
Brilliant, and thank you so much for inviting me. It's a real shame that I can't actually come in person, but obviously we are going through a global pandemic right now. And indeed, over the last year, this is the main thing that's been concerning us, the impact of a global pandemic. But in reality, this is not the only challenge we are facing. So if we look at temperatures, we can see that average temperatures in 2020 were around 1.2 degrees Celsius above pre-industrial levels. And in fact, like many years before it, 2020 was one of the warmest year on record, a third warmest year after 2016, 2019. And in particular in 2020, temperatures were very high around the Arctic. And this is despite the fact that, as you remember, 2020, we were under national lockdowns and reduced aviation. So despite all the international agreements as well, 2021 is projected to have a massive increase in levels of emissions as some countries are overcoming the impacts of the pandemic. So what does this actually mean? So as temperatures are rising across the world, as patterns of precipitation are changing, conditions are becoming unsuitable for many organisms. And this is something that's already been forcing many organisms to shift their ranges in pursuit of suitable conditions. So why is it really a problem? Well, not only is something that's going to have detrimental effects on biodiversity, but it's also something that will impact the ecosystem services they provide. And as a result of that, our food security and really our survival in general. But we know that climate change is not something that's happening on its own. It's happening over this backdrop of very intensive anthropogenic land use changes that results in habitat loss, degradation, and fragmentation. So how biodiversity will respond to these interacting stresses really depends on, first of all, the extent of these changes, so their magnitude and how fast they're happening, but also it depends on the intrinsic factors for the organisms themselves. So their adaptive capacity, their diversity and diversity of responses, potential for phenotypic plasticity in responding that way, but also the dispersal ability and ability to move away from these changes. So commonly, in order to understand the vulnerability of biodiversity to these global environmental changes, we use correlative modeling approaches like species distribution models, also known as ecological niche models. So how these approaches work, they use information on where the species is found, so location records, in combination with environmental conditions in these locations, to project where suitable conditions may be found across a wider geographical area. And they can be combined with scenarios of projections of future temperature changes and changes in patterns of rainfall to predict where suitable conditions will be in the future under climate change. This kind of approach has been used extensively. For example, in a really seminal paper from 2004 by Chris Thomas and colleagues, they predicted that 18 to 35% of species will be committed to extinction by the end of the century. And the range depends on the range of emission scenarios. But these kind of approaches have been criticized for being oversimplistic and really disregarding quite important mechanisms that affect the impacts of future changes, as well as the response of species to future changes. And these include evolutionary mechanisms, and in particular adaptive variation. Also physiological aspects and thermal tolerance and capacity for physiological responses, demographic factors, dispersal ability and barriers to dispersal, and finally, species interactions. So my research really aims to address this gap in our ability to assess vulnerability to environmental changes. And we do it in my group through using integrated approaches. And by that, I mean combining genomic data with ecological research, geographical data, and different modeling approaches. But also we don't look at one threat on its own. So we don't look at climate change on its own, but we look at the interactions between climate change and anthropogenic land use changes. Now, a lot of our work focuses on bats. So why bats? Well, they're very species rich, 20% of mammals with over 1,400 species. They're distributed pretty much everywhere across the globe with the exception of the Arctic and Antarctica. So if we look at them and we're particularly interested in response to environmental changes, we can see a variety of responses due to their wide geographic distributions. Also, bats play important roles in the ecosystems through seed dispersal, pollination, forest regeneration, and control of insect pest populations. We also know that a high proportion of bats are under threat, and many species that are even relatively common have experienced population declines. But beyond that, going back to the situation that we're currently in the global pandemic, we know that bats are hosts of many zoonotic pathogens, including coronaviruses. So it's very important to know how the distribution may be changing as a result of climate change and anthropogenic land use changes. Now, in terms of bat sensitivity to climate change, bats are thought to be relatively sensitive due to their very large membranous wings, which means they have large surface area relative to their volume. And as a result of that, they can lose a lot of water to evaporation. And studies that have been carried out in desert environments have shown that bats over there have physiological adaptation for reduced evaporative water loss rates. And that means that under changing conditions, as conditions become warmer and drier in parts of the globe, bats will require physiological adaptations to be able to survive. And we've seen over the last few years mass mortality events, in particular in Australia, following heat waves, with in particular flying foxes, pteropodids, just dying in thousands in response to the heat waves. But this is something that's also been seen in insectivorous bats in Southeast Asia, and also something that's affecting bats across the Mediterranean. So today I'm going to show you how in my group we try and integrate these kind of approaches to improve the way we are able to assess vulnerability to global environmental changes. So I'm going to focus on integrating adaptive responses, dispersal and barriers to movement, ecological interactions, and impacts of multiple stresses. I'm going to finish by introducing a framework that we developed to try and assess responses to environmental changes, in particular climate change, and show you how this kind of framework can also be applied to inform conservation management. So I'm going to start with looking at adaptive variation and how we can use genomic approaches to understand adaptive responses to climate change. Now, currently, these kind of correlative modeling approaches, they treat species as a single unit across their whole range. Now, we know that this is not likely to be the case, in particular when we think about responses to climate change, because individuals, for example, in this bat Myotis dobentoni that are found right here in the south of Spain, are adapted to different climatic conditions than those individuals that are found here in Sweden, for example. So we developed approaches to directly integrate adaptive variation into our assessments of range losses under climate change. And we test this approach using a set of cryptic bat species, Myotis escalari and Myotis crypticus. These are two Mediterranean bat species. Myotis escalari is restricted to Spain and Portugal, the Iberian Peninsula, while Myotis crypticus is found in the northern parts of the Iberian Peninsula, southern France, and Italy. We generated genomic data sets for these bats using DD-RADSIC, so a reduced genome representation approach. and ended up with a data set of around 20,000 SNPs, single nucleotide polymorphisms. So the first step is to identify climate adaptive individuals. And we do it through using genotype environmental association analysis. So this is a kind of approach that relates allele frequency or genetic makeup to environmental variables. Now, in our case, we focus on two primary environmental variables, maximum temperatures and summer rainfall. And the reason for that is because these are variables that are likely to change dramatically under climate change, in particular around Mediterranean habitats. But also these are variables that are very important for bat survival and reproductive success. So I've already mentioned the mass mortality events of bats following heat waves, but also in terms of water availability, bats reproduce during the summer. These are insectivorous bats, so they rely on insect availability. And insect availability is linked to water availability in Mediterranean habitats in the summer. So changes in these parameters will affect really bat survival and reproductive success. Okay, so we used our genotype environmental association analysis approach, looking at these two environmental variables of interest, and we managed to identify 32 and 38 genomic regions, one in each bat species, associated with maximum temperatures and summer rainfall. Then we plot to see where individuals fall along the ordination space relative to these two climatic gradients of interest, based on their genetic makeup in these SNPs that are climate adaptive SNPs. And we divide our individuals. There are those that are associated with hot and dry conditions, here in white, versus those that are associated with cold and wet conditions, here in grey. And we remove the intermediate individuals. Then we directly incorporate that into our models. So the models you see here on the left were generated using all the location records for the species. In the top, we have models based on present climatic conditions. In the bottom, projected climatic conditions for the end of the century using their more severe emissions scenario. Now, in these maps that you see here, blue colors represent unsuitable areas and orange and yellow colors suitable areas. So if we compare the maps of the present to the future for this bat Myotis escalari, we can see extensive losses of range suitability in the Iberian Peninsula, which is where the species is restricted to. Now, the models that you see on the right were generated separately for bats that were identified as associated with hot and dry conditions in red versus bats that have genotypes associated with cold and wet conditions in blue. And yellow, we have areas of overlap. So if we compare the top map present conditions versus the bottom map future conditions, we can see that we still see range losses under climate change. However, when we look at these two bottom maps, we can see that when we incorporate genetic variation, adaptive variation into our models, the range loss projections are reduced substantially. Now, the same situation for the other bat species, Myotis crypticus. But what's interesting to see in this bat species, if we look at this bottom right figure, is that we see an expansion of their hot, dry adapted genotypes, so the red genotypes, on the expense of the blue genotypes, so the cold, wet adaptive genotypes. So we're seeing not only that when we incorporate adaptive variation, we improve our projections of what range losses may be like, but also we can see how their composition of genetic variation is going to change in our species. So other approaches that we're using to look at impacts of, to try and incorporate adaptive variations in our assessment is to try and understand adaptations to arid conditions. So my PhD student, Evie Morris, is looking at adaptations to aridity in the bat Pipistrellus cooley, or the coarse pipistrelle. Now, this bat's a common Mediterranean bat species. But recently, over the last 60 to 70 years, this bat has expanded its range in Israel to desert habitats where it wasn't previously found. And this is following the expansion of human settlements and agriculture in the area. Now, this bat not only expanded its range to these areas, it's really thriving there, and it also managed to reach these hyper-arid desert areas and survive there, all the way up right to the south. So what Evie is doing, she's looking at two aspects. First, the bat genome, so the host bat genome, to see if we see any signature of adaptive variation in response to this quite recent expansion into desert environment. She's also looking at the gut microbiome of these bats to try and see whether we're seeing any changes in the gut microbiome. Now, it's thought that the gut microbiome will be able to respond much faster to environmental changes than the host itself. And therefore, it may offer the host a way how to facilitate response and acclimatization, at least, to warmer and dry environmental conditions. Okay, so let's start with the bat DNA. So Evie collected samples for 102 bats across their aridity gradient. And she used DD-RUD sequencing to generate a genomic data set of more than 40,000 SNPs. And then she used a combination of two genotype environmental association analysis methods and identified 44 genomic regions or SNPs that are associated with adaptations to maximum temperature and aridity. Then, as before, she plotted where the individuals fall along their ordination space relative to these two environmental gradient based on the genomic makeup in these 44 SNPs. Now, in the maps that you see here, we color-coded individuals based on where they originally came from. So in red, we have the individuals that came from the hyper-arid areas from the Rift Valley. In yellow, we have individuals that came from arid areas, and in blue, we have individuals that came from the Mediterranean environment, so the more mesic environments. While in gray, we have the intermediate areas, so it's semi-arid areas at the edge of the desert. And what we can see here, that when we look at adaptive variation, we're already seeing a signature of the split of the different populations here, with hyper-arid bats very closely associated with the hot and dry conditions, while the more mesic bats are associated with much wetter conditions. So we see a very distinct population structure here separating these bats. Now, what's interesting is that when we look at how the population structure based on neutral genetic variation, we don't see any population structure whatsoever. And in fact, all the individuals just fall into one single cloud in the middle. And individuals that come all the way down from the most hyper-arid areas in the south fall right near individuals that come from the more northern mesic parts. So what we're seeing here is population structure that's based on adaptive variation rather than just neutral genetic variation. And this is likely because we still have ongoing gene flow coming from the more northern areas, the more mesic areas into the desert. Okay, so next let's look at the gut microbiome. So this is using DNA metabarcoding approaches, but Evie is now working also on shotgun metagenomic approaches to supplement this. So if we look at the composition of the gut microbiota of individuals that come from arid environments in yellow versus Mediterranean environments in green, we can see that in arid environments, we have a higher proportion of phemericutes, and these are associated sometimes with increased efficiency of extraction of energy from food and storage. But we have a lower proportion of actinobacterioids and cyanobacteria. But if we look at the overlap in the gut microbiota of these bats, that those that come from arid environment versus those that come from more mesic environment, we see that overlap between them is not different from random. However, when we compare overlap in the gut microbiome of Pipistrellus cooley bats in arid environment to those of desert obligate bat species, Hypsigo ariel, we see that niche overlap in the gut microbiota is significantly higher than random. So we see that the gut microbiome of the bats that have expanded into desert environment has become much more similar to that of desert obligate bat species. So put this together, we're seeing already responses to this expansion into a desert environment, whereby the bats itself are showing adaptive variation that's associated with arid environment, but also the gut microbiome has become more similar to that of bats that are found in arid environments. Okay, so next I want to move to incorporating movement and dispersal. Now, the reason why it's important to think about this is because the ability of organisms to simply shift the range and reach climatically suitable areas is likely to be affected by geographical barriers or landscape barriers to movement. And these can be either natural barriers or anthropogenic barriers. But commonly, this is something that's entirely neglected in species distribution models. And where it has been included, it's mainly been included through creating dispersal kernels, so limiting the distances a species may be able to reach. And the problem with that is that it totally doesn't look in any way at the impacts of the landscape and barriers to movement. So instead, what we use the landscape genetics approach to relate spatial genetic patterns to the effect of the landscape on individual movement. And we use this approach to look at the impacts of spatial scale, for example, on landscape connectivity and how it affects genetic connectivity. So here is an example, the Placotus austriacus, the grey long-eared bat. We saw that in the south of England, the loss of unimproved grasslands in the past 100 years as a result of changing in farming practices has resulted in population fragmentation and reduced genetic connectivity in these bats that are closely associated with this habitat type. We also use this approach to see the impacts of landscape connectivity on range shifts. So we asked, how can individuals that are found in areas that are currently climatically suitable, but will become climatically unsuitable by the end of the century, be able to reach these areas that will retain climatic suitability, here in black? And we generated these range shift corridors based on the effect of the landscape on current patterns of genetic connectivity. And we can use this to identify pinch points, so areas where landscape connectivity is likely to limit the ability of organisms to shift the range. We also use this approach to look at the effect of landscape barriers on the potential for evolutionary rescue. And when I talk about evolutionary rescue, what I mean is the spread of adaptive variation into maladaptive population, and therefore maladaptive population may be able to survive. So what we're talking about here is populations that are adapted to cold and wet environmental conditions. So going back to our example of the Myotis escalari and Myotis crypticus, the two bat species in the Iberian Peninsula, these are two forest bat species. So when we look at the effect of landscape on genetic connectivity between their populations or sites, what we find is that they're mainly affected by forest variables and topographic variables. So then we take this information forward to project what will be the potential for movement, or in our case, what we're interested in is gene flow, between populations that are adapted to hot and dry conditions in white, to those populations that only have individuals that are adapted to cold and wet conditions in grey. And these maps show the potential movement density given the effect of the landscape on genetic connectivity. So yellowish greenish areas represent high potential movement density, while blue areas represent low potential for movement density. And we can see that adaptive variation is likely to reach some of their sites and the populations. However, others will remain more isolated. And what it shows that although evolutionary rescue is possible, it's likely to be affected by landscape connectivity. Okay, moving now into ecological interactions. A lot of my work looks at trying to identify the effect of ecological interactions on species distributions in order to understand what we need to actually incorporate in our models and how to do this. So my PhD student, Roberto Novella Fernandez, who recently submitted his PhD and currently doing a postdoc in Germany, worked in his PhD on trying to infer the role of competition in shaping species ranges. So it's commonly agreed that competition has a strong effect on local scale distribution, local scale habitat use of species. However, it's usually thought that the broad scale distribution or the range of species is more affected by climatic conditions. So what Roberto did, he developed a measure of geographic avoidance to assess the effect of competition on species ranges and whether species tend to geographically avoid one each other or not. So he applied this measure to 4 sets of cryptic bat species. Now we're interested in cryptic species because they're likely to be ecologically similar and therefore have higher chances of competing with one each other. And first he just used species distribution models to look at their range. And in these maps, we can see unsuitable conditions for this species in grey, while areas that are predicted to be suitable for the species are marked in colours. So in green, we have areas that are predicted to be suitable for the species by the model, and they're actually realised by the species, versus orange areas represent areas that are predicted to be suitable, but are not realised by the species. And what we're really interested in looking at the extent of overlap between these two species in areas that are realized versus areas that are unrealized. So the results of the geographic avoidance measure were compared to 100 null models generated using virtual species in order to identify those pairs of species where levels of geographic avoidance are significantly higher than random. And Roberto has identified two pairs of species, the Eptesicus group and Plecotus austriacus and Plecotus colombatovici, where levels of geographic avoidance across their ranges is higher than random. Then he looked at levels of ecological similarity between the different pairs of species and whether there's any evidence of resource partitioning. So when we talk about ecological similarity, we talk in terms of habitat, diet, and roost use. And interestingly, what he found is those pairs of species that show high levels of geographic avoidance are those that don't have any fine-scale mechanisms of resource partitioning and are highly ecologically similar to one each other. And what this suggests is that broad-scale patterns of geographic avoidance emerge in the absence of fine-scale mechanisms of coexistence. So then we took us forward to try and understand what happens actually with the range suitability of these bats under climate change, and how would this geographic avoidance likely to affect the future ranges. So we look at these two pairs where we know that they tend to geographically avoid one each other, and projected their ranges under future climate change. The areas that are marked in black represent areas where the future overlap between the two species. Now, what we can see for the first set, the Eptesicus set, we see reduced levels of range overlap under future climate change. However, for the Plecotus pairs of species, we see extensive increase in range overlap. And in particular for Plecotus colombatovici, much of its future predicted suitable range will overlap with that of its competitor. Now, that meant that the species is not necessarily likely to be able to expand to suitable ranges or even survive in areas that will remain suitable as a result of competitive interactions with a competitor that it's currently avoiding in its distribution. And what this really shows is the importance of trying to incorporate biotic interactions in our assessments of future range suitability, because some areas that we predicted to be suitable may actually be areas that the species is not able to occupy due to other species that it's competing with already existing there. Other approaches that we're using to look at biotic interactions is through DNA metabarcoding to sequence the diet of insectivorous bats. And what we're interested in is identifying fine-scale mechanisms of resource partitioning. But also we looked at bat communities across a wider geographical scale. So we looked at 7 cave-dwelling bat species across the whole of Europe, across different environmental gradients, to try and link their diet with environmental gradients. And what we find is that those bat species can be divided into 4 main clusters or groups based on their diet. But then we combine this information with species distribution models. And what we found is those species that have broader trophic niches, so they eat a more varied types of prey, are also the species that have broader environmental niches, so they found under more variable environmental conditions. And this is a really nice link between trophic ecology and species distribution and the environmental conditions that they can be found under. Okay, I'm going to move now to talk about the effect of multiple stressors and how we can integrate in a way the impacts of climate change and anthropogenic land use changes. In this study, we focus here on the Ethiopian highlands and a specific bat species in the Ethiopian highlands, Plecotus balensis, the endemic long-eared bat species. Now, the reason why we're interested in this bat species in particular is because this bat species is found at very high elevations, so in the Afro-alpine and high Afro-montane habitats. And we know that these kind of habitats are the habitats that are likely to be very sensitive to future climate change and experience disappearing climates. We're also particularly interested in the Ethiopian highlands because Ethiopia itself have undergone very extensive changes in the past 150 years. This is following a very rapid human population expansion from around 6.6 million to more than 100 million. And this has resulted in extensive habitat changes, habitat loss and degradation, and something that has impacted biodiversity there. So together with Javier Juste, we went on 3 different expeditions to the Ethiopian highlands to sample these bats across 5 mountain ranges, 2 at the southern Ethiopian highlands and 3 at the northern Ethiopian highlands, separated by the Rift Valley. So first, we used distribution models to look at the effect of climate change past and future on habitat suitability for these bats. And this is a distribution of suitable conditions for these bats under present conditions. In those maps, orange and yellow areas represent suitable conditions and blue and greenish unsuitable conditions. And we can see here that quite limited suitable conditions exist for these bats and primarily limited to these much higher altitude environments. Another interesting thing to see here is that we have the Rift Valley here that's really separating the 2 areas of suitable conditions for these bats. And indeed, when we look at the genetic data, it really mirrors it. So those individuals that were sampled south of the Rift Valley are here in yellow. Those that were sampled north of the Rift Valley are in green. So they split very strongly, very distinctly into 2 separate populations. Okay, then we projected or hindcasted the distribution of suitable conditions based on conditions during the last glacial maximum around 21,000 years ago, when conditions were much cooler there. And as a result of that, extent of range suitability was much larger for these species. And in fact, the past suitable range was 353% larger than current suitable range. We also projected it to suitable conditions in the future by the end of the century using the more severe emission scenario. And we see extensive range loss predictions, with future ranges being 74% smaller than current suitable ranges. So what we see here that suitable range for these bats have been gradually decreasing in the past few thousand years, and is projected to even decrease much more extensively under future climate change. Next, we looked at the effect of anthropogenic land use changes. And we used approximate Bayesian computation model-based inference of demographic history to see if we see any signature of population changes in these bats in response to the land use changes, and to try and also time when these changes may have happened. Now, the model that received the maximum posterior probability support, so 0.99, is one showing a 6-fold population decline in the southern population in the past 150 years. Now, if we remember, 150 years correspond to the period where human population started to expand very rapidly and extensive land use changes have occurred. We also looked at the impact of agriculture expansion on genetic diversity. And what we found is that allelic richness or genetic diversity decreases as the percent of arable cover around the population or around the capture site increases. So the link here between amount of agriculture area, land converted to agriculture, and loss of genetic diversity. So putting all of this together and thinking about a wider context, what this study really highlights is the threat to Afro-alpine and Afro-montane species. That on the one hand, they're threatened by disappearing climates due to climate change and loss of the range suitable range for them. But on the other hand, many of them are already experiencing very extensive loss of genetic diversity and population decline as a result of land use changes. Okay, I wanted to finish by telling you a bit about an integrated framework that we developed to try and combine these different aspects when we assess vulnerability to climate change. And we ask here which populations are under threat to climate change. And we answer it using a combination of genomic approaches with ecological research and different modeling approaches. So we look at 3 factors here. The first is exposure. So changes in climatic conditions, how extensive are they likely to be where the species is found. The second is sensitivity. And we're really asking, can they actually cope with future changes? And what we're talking about here is in terms of their genetic makeup and adaptive genomic variation. And third, we look at range shift potential. So can they actually move away and reach climatically suitable areas? And we test this framework on the grey long-eared bat, Plecotus austriacus. Now, the spat is relatively common around the Iberian Peninsula, but is very rare in northern parts of its range, like the south of England. So we sample populations of this bat across the climatic gradient, all the way from the Iberian Peninsula, coming up to southern England. So first looking at the exposure and will they be affected? So we use species distribution modeling. First to look at the distribution of suitable conditions for the species under present climates. And we can see that indeed much of the Mediterranean area is highly suitable for this bat. And in fact, all the populations that we sample are found under suitable conditions. Then we projected to conditions towards the end of the century using the severe emission scenario. And we see extensive range losses projected around the Iberian Peninsula, while we see a northwestern shift in suitable conditions, with much of Britain becoming highly climatically suitable for these bats. And in fact, we identified 3 populations that are currently found under suitable conditions, but will be found under unsuitable conditions by the end of the century. We also looked at the extent of environmental dissimilarity between present and future conditions. And we identified these 2 populations that are found in areas where maximum temperatures are predicted to increase by over 8 degrees Celsius. We also identified these 2 populations that are found in areas where summer rainfall is predicted to decrease by over 50%. Okay, so next we look at sensitivity and can they cope? As I said, when I talk about sensitivity, I talk in terms of adaptive genetic variation. So we use similar approaches as before, the genotype environmental association analysis, to identify adaptive variations associated with maximum temperature and low summer rainfall. And then we identify those populations that have low levels of adaptive variation associated with these conditions that are likely to be more prevalent under future climate change. Therefore, these populations are likely to be more sensitive to future conditions and future changes. And then we looked at movement potential and can they get away? Can they actually shift their ranges? So once again, we use the landscape genetics approach to try and identify the landscape variables that most affect genetic connectivity or gene flow between populations. Then we plot the potential movement density based on these conditions. So movement density potential ranges from high potential in yellow and low potential in blue. And we can see that under present conditions, all our populations in the Iberian Peninsula are nicely linked to one another, and also some gene flow occurring all the way up to England. Now, if we look at what will happen under future conditions, what we can see overall is reduction in landscape connectivity. And we can identify these 4 populations that will become isolated under future conditions, meaning that they won't easily be able to shift their ranges. Quite worryingly, another thing that we identify is actually reduced connectivity out of the Iberian Peninsula as well. Okay, so now let's put everything together. And what we're looking at here is, in a way, a risk assessment framework, trying to divide our different populations based on the level of vulnerability. So what I'm going to show you here, on the left, we have the least vulnerable populations ranging on to the most vulnerable populations on the right. So let's start with the left-hand side, the least vulnerable populations. Now, these are populations that are found in areas that are likely to retain climatic suitability in the future. They also already have adaptive variation that's associated with warmer and drier conditions. And they're found in areas that will retain landscape connectivity in the future, meaning that they can shift the range if they need to. Now, on the other end of the scale, the highly vulnerable populations, we have those that are found in areas where climatic conditions will change dramatically and become unsuitable for the species. These are populations that have high sensitivity to future changes because they don't have adaptive variations that is associated with warmer and drier conditions. And also they're found in areas that will become isolated under future conditions, so they won't easily be able to shift their range. Now, in the middle here, we have these populations found in areas where climatic conditions are likely to change. However, they either have lower sensitivity to these changes because they already have adaptive variations that's associated with warmer and drier conditions, or they're found in areas that will retain landscape connectivity and therefore they'll be able to shift their range and reach other climatically suitable areas. Now, we can use this kind of information to inform adaptive management recommendations under climate change. Now, the conservation recommendations that I'm going to give here range from the least intensive intervention to the most intensive intervention on the right. So here, if we look at the low risk or low vulnerability populations, because they're found in areas that are already, they will retain their suitability, and they're able to cope with these changes, or likely to be able to cope with these changes because of their adaptive variation, what we really want to do is bolster these populations through increasing habitat suitability for them where they're found. So this is to ensure that we'll have large populations there in areas that are protected and will be able to survive there in the future, and perhaps also be able to generate these gene flow of adaptive variation that will reach other populations. Now, if we look at the other extreme, the most vulnerable populations, they cannot stay where they are, but they cannot shift their range on their own. So the only solution in a way is assisted translocation, to physically go there, lift them up and move them to somewhere else. Now, although this is a viable management option, if we think about the extent of management that will be required under climate change, it's not really feasible to think this is something that can be done for most species. So instead, what we may want to focus is more on the mid-vulnerability populations, where if we increase landscape connectivity, we can facilitate range shifts as well as facilitate the movement or gene flow and the spread of adaptive variation between populations. Now, this is a kind of management intervention that's likely not just to benefit this species, but likely to benefit many other species as well. So I'm going to finish with some take-home messages. I hope I managed to convey to you the importance of using integrated approaches, and in particular integrating genomics data and ecological information into our modeling approaches. I also hope I managed to show you the importance of considering multiple stressor and looking at the effect of multiple stresses together. Climate change is not happening on its own, it's happening over this backdrop of very intensive and extensive habitat changes. So we must try to integrate these two impacts together when we assess vulnerability to future changes. I showed you that current models may be overestimating vulnerability to climate change, and in particular overestimating range losses because they disregard the effect of adaptive variation. And we showed that how it's important to actually incorporate adaptive variation into our models. Now, the problem here is that what we end up is misguided conservation efforts because our models are not actually predicting what may happen. I also showed you the importance of landscape connectivity, first of all to facilitate range shifts, but also to facilitate evolutionary rescue. And this is why it's very important to incorporate landscape connectivity in our models and better understand the effect of landscape on gene flow and movement of organisms. And finally, I showed you the effect of ecological interactions, where they don't just affect fine-scale resource use, but also species ranges, and therefore they should be incorporated into our models assessing range changes under climate change. I want to just end by thanking the many collaborators and funders that funded these projects and collaborators that helped with carrying out all the work, and particularly my two PhD students that did quite a lot of this work. I also wanted to thank you for listening, and I'll stop sharing.
Thank you very, very much, Orly. This was I am astonished by all the integrating, all the stuff that you have integrated in a very coherent framework. It's really amazing. While the audience comes with new questions, I still have some. I have some of my own. One of the things that struck me the most was how you can create, compile these maps of density of movement, density of probability of movement, mm-hmm. and how can you extrapolate them or create predictive models for the future? Like, could you give us a little bit more detail on that? Like, for example, what are they based on apart from genetics? Environment, I don't know.
Yeah, so I obviously flew through it a bit, but I'll rewind back a bit and describe the process. So we start by looking at which landscape variable affects genetic connectivity, so gene flow or genetic differentiation between populations. Mm-hmm. Now, what we assume is that genetic gene flow tells us something not just about movement, but but also about movement that results in reproduction. So it's more the fact, if we're thinking about population establishment, this is more important than just whether they can move across the landscape. Now, how we do it, we look at a variety of landscape variables that we think may affect the species. We create these cost layers that, based on expert opinion, of what we think may be the cost for the species crossing these landscape variables. And then we use a statistical framework to compare these different landscape cost surfaces to our matrices of genetic variability or genetic distance between our individuals, and identify these landscape variables that best explain patterns of gene flow that we're currently seeing. Now, once we identify these landscape variables, we also have the cost of the individuals crossing these landscape variable, and we create these maps that actually show the cost of crossing the whole area between our different populations. So this is what you see, the movement density maps are actually, it uses circuit theory. So it sort of sends out electric circuits across all directions and see which one of them are resulting in areas where the cost of crossing the landscape is lower for the species.
By the amount of genetic information shared, I guess.
Yeah, so the amount of genetic connectivity or gene flow between these areas. So obviously areas that will have high gene flow and high connectivity, they're likely not separated by any landscape that form a barrier for the species.
Right.
While if you have two populations that you have very limited gene flow, they're likely to be separated by something that may stop the individuals from being able to cross this area.
Okay, so I have a couple of questions from Javier Juste. He says, "Great talk, Orly." He has, the first question is regarding Pipistrellus cooley study. Do you have any idea about how reversible is this aridity-driven local structure of the genetic makeup of the species? Shall I repeat?
Yeah, unfortunately, no. So all we know is what we're seeing. And we've only used, in a way, correlative approaches to relate the genomic makeup of these bats to the environmental conditions where they're found. What's really necessary is to get into it in much more depth and really try to look at whole genome studies and try to understand what mechanisms are involved in their adaptations to these environments. What I am assuming is that may have happened is that only those individuals that already have the genetic variation that's more associated with warmer and drier conditions are those that were able to survive in these deserts, because we are seeing continuous gene flow still occurring from more Mediterranean populations into the desert populations. So this is happening, and this is adaptive variation, local adaptations are happening despite the extensive gene flow that's happening. And this is likely due to the very strong effects of selection in these environments. So whether it's reversible or not, this is quite difficult to know, and especially because it's something that's just happened so recently. The expansion into the desert only occurred in the past 60 years or 65 years.
Right. Javier Juste was also interested in whether sorry, the questions are growing and moving the lines. So whether the adaptation would be, in the process of adaptation would be implying in losing plasticity for individuals.
Yeah, very good question. So this is another thing that we're quite interested in, is looking at their plasticity and plastic responses to changes. And it's definitely something that needs to be looked at, because adaptation can happen in different ways. But one way that it can happen when you have very strong, rapid adaptive responses, you can result with a loss of phenotypic plasticity, which will affect the ability of the species to respond to environmental changes as well. So it's quite interesting to look at the interface between these two mechanisms. And we do have a grant proposal that's in review right now that's looking at phenotypic plasticity and looking at using RNA-seq to look at differences in gene expression between bats that are found in those more desert conditions versus mesite conditions, as well as looking with those that are found in desert conditions between the spring and the summer. So whether we see these really differences in phenotypic plasticity happening, and to try and understand what is really the mechanism here that's driving the adaptive responses to the desert conditions.
Thank you, Orly. I have another couple of questions from Maria Jose Ruiz. She says, I have two questions. Thank you very much for your great talk. In the models to predict evolutionary rescue, what landscape parameters did you use? Are they current landscape features or predictive?
Yeah, so in the case of these bats, what we used is current forest cover. So two aspects we found that affect gene flow between current populations of these bats, and these were forest cover and topography. So topography is not something that's likely to change, so this was fine. In terms of forest cover, we just kept current forest cover. Now, the main reason why we did it is because those maps that we do have, where we project for projected future land cover changes, they're actually done through downscaling 50 by 50 kilometer cells. So the level of accuracy at the smaller level is quite reduced. The other reason is because it's likely that we're talking about quite small timescales, right? We're talking about 2070. And it's likely that forest composition is not necessarily going to change as dramatically within this period. Obviously, if we look at much further times, we're likely to see quite extensive changes in forests, and especially with difference in patterns of recruitment and species compositions, and as well land use changes. But we are using current conditions because of that, because of the lack of availability of very detailed future projections.
On that line, thank you very much. On that line, I would like to add the following question that remained before, which is from these variables that you use to create the movement density maps, where the climate, how important were the climatic ones, the ones that allow you to make predictive models for the 2070, etc. Were they very important?
Yes, so so these were quite important climatic variables, yeah. And for example, for the case study that I showed in the end, when we looked at movement density and projected it to the future to see how it will affect connectivity and the ability of populations to shift the range, we used the variable that came as the landscape variable that affected genetic connectivity the most was the species distribution models themselves, so habitat suitability based on these models. And this is why we were able to project it to the future and see as habitat suitability changes, how is it likely to affect genetic connectivity as well in these bats. So climate did play quite an important effect in these models.
Okay. Thank you again. So there is another more question from Justine Levela. I hope I said it well. So she congratulates you for your amazing work again, and she says, did you have some demographic data to include them into some evo-eco models to test the hypothesis of evolutionary rescue, for example?
Yeah, interesting point, and I would love to do that, definitely. And collaborating with people from the University of Aberdeen that developed Rainshifter to try and create these kind of models that incorporate a bit more demographic data as well as incorporating genetic and genomic data into them. So to try and understand really how would species actually shift their ranges. So now we're just projecting, yes, this is likely to be the distribution of suitable conditions. But what we really need to include into this is how are they actually likely to move across the landscape, given what we know about demographic data and what we know about adaptive variation. So this is something that we're currently trying to develop with them.
Great. Another question from Maria Jose Ruiz Lopez is, regarding the framework that you present, do you think it could be easily used for other species, such as mosquitoes, where climate change is predicted to have a strong impact on range shifts?
Yes, so definitely, and I truly hope so. So I'm involved in a project led by somebody in IDIV that will be looking exactly at that. So we're going to be collecting from our online databases loads of available whole genome sequencing data and RAD-seq data, and try and run this framework on as many species as we potentially can. The original focus is on mammals and reptiles primarily, and across Europe and Africa. However, we're going to create these pipelines that will enable this framework to be run on as many species as you can think of. It all depends on data availability, obviously.
Thank you. So Carles Villar says, thank you for a very nice presentation. And he says, if I understood, sensitivity was assessed by looking for genomic variants associated with environmental factors. However, do these same environmental factors play a key role in habitat selection models? Do you restrict sensitivity analysis to variables important in niche modeling?
Yeah, so as it happens, these variables are actually important in the niche models for these bats. And as I mentioned, the reason why we focus on these variables is not only because these are variables we know are going to change dramatically, in particular around Mediterranean habitats under climate change, but also because they're so important for bat survival and reproductive success. And in fact, these variables, in many of the models that we run across many bat species across Europe, they come as some of the top variables, in particular for more Mediterranean bat species. So summer rainfall is very important for a lot of Mediterranean bat species.
Summer range, okay. So Ivan Gomez Mestre says, hi Orly, great talk, thanks. And have you been able to test the adaptive value of the observed genetic differences experimentally, or is it based solely on correlational inference? It's only based on correlative approaches. So one of the downfalls, in a way, of working with bats is that they're very long-lived. Also, most of them are protected species, and we can't simply take them into the lab, manipulate them, and assess a lot of these impacts. So what we are using instead is the more correlative approaches. I've been to a very interesting talk, however, as part of the BAT1K project, with somebody talking about using cell lines to be able to actually extract this kind of information and create a lot of lab manipulation just on cell lines that have been extracted from simple wing biopsy punches. So having minimal impact on the bats themselves, but from that we can use a bit more experimental approaches. So this is something I'm really looking forward to seeing more how it's going to be developed and how it's going to be used. That sounds very interesting. Okay, and finally, Irene Mendoza says, thanks for such a great talk, Orly, and she was asking for the trophic interactions in relation with the environment. Have you seen any shift or range? Have you seen any shift of range because the prey of the bat is not present anymore? Yeah, very interesting question. And I would say a lot of my work on biotic interaction really focuses on competition. However, biotic interaction when it comes to trophic ecology are in particular important, perhaps even more important, because if what you eat is not there, you will not be able to survive. However, in the terms of bats, I think it's perhaps not quite as critical as it is for other taxa. And this is because bats are very generalist. So even those that we call specialist bats, they specialize on Lepidoptera, right? But they don't specialize on a Lepidoptera species. They will specialize on any Lepidoptera that's available within some limits of palatability and size. So what we have here is a situation where as long as invertebrates are there, as long as Lepidoptera, for example, is there, then bats will have something to eat. What's likely to affect bats a lot more is actually global decline in insect abundance. And this is something I'm very much interested in. And I think that's something that we need to try and incorporate better into our models, because this is likely to have a very major impact on insectivorous animals in general. Super interesting. While we have to give a little more time to pile up a few more questions, I wanted to ask you too also about how do you do to obtain a nice number of location data for cryptic species? I saw in your plots a lot of actually measure individuals, which is nicely surprising for a genetic study. But how do you do it? How do you find these cryptic species? And well, I would have to thank for that Javier Jose and Carlos Ibanez from Doñana for their extensive work across the Iberian Peninsula. And a lot of these samples have been collected by them previously, and actually part of the collection that's in Doñana, as well as carrying out field work and working with a lot of collaborators. So a lot of the projects I have involve collaborators from across many countries, because I see it as the most viable way, really, how to get sufficient samples to really represent these climatic gradients. Yeah. And this is something we really need. If we're trying to look at very fine-scale changes and fine-scale differences, we need the sample sizes, and we need to be able to cover all these areas. So yeah, it's thanks to not me, but many other people's work for many, many years collecting these samples. That's very true. It's the time for assembled large groups from across geographic scales. Thank you very much, Orly. I don't see any new question here in the YouTube chat. So, do you have any message for us? Any final words, as we said?
Well, I just wanted to thank you for inviting me, and I hope that next time I'll be able to come in person.
That will be absolutely great. Okay, so please stay with us while we close this seminar, so we can comment a little bit more afterwards. And for everybody who is looking, the next Wednesday, day 2, we will have our next seminar about bears in urban environments, which is given by Jesus Manuel Diaz Fernandez. And just remembering you that it won't be Thursday 3 because that day will be holiday. So we will have it same time, same channel, but in day 2, Wednesday. So I hope you all enjoy the show and see you next week. Bye-bye.